of this past paper is related to meiosis. Let me read out a part. In sexual reproduction, meiosis occurs to produce haploid gametes from a diploid cell. So when we talk about meiosis, definitely we are talking about the formation of the haploid gametes from a diploid cell. Explain. Now here our command word is explain. When we have command word explain, again, we have to explain certain concept in a very comprehensive way. It can be in the form of points or it can be in the form of a paragraph, but make sure that when we explain, it should actually cover the important aspect of the terminologies given or the processes given, etc. For example, here in this case, explain the meaning of the terms haploid and diploid. Apparently, it's not a very complicated thing to explain because haploid and diploid, many times we keep talking about them whenever we discuss the meiosis process or whenever we discuss the inheritance topics. So you simply have to talk about what is haploid, what is diploid. So let me go directly. Haploid, as you know, haploid means half number of chromosomes, or you can say n number of chromosomes, or you can say one set of chromosomes, and this we only find in the gametes or sex cells. Diploid, double number of chromosomes, or 2n, or two sets of chromosomes, or definitely we find in the normal cells or somatic cells. So basically you just have to say one line for haploid and one line for diploid, and it can be any alternative wording. AW means alternative wording, but make sure that you are telling the examiner the exact information about haploid and diploid. You do not need to write the details about haploid and diploid, but you just need to focus what is haploid and what is diploid, as the marks are only 2. So it means you will be getting 1 mark for haploid and 1 mark for diploid, and that's it. Okay, now let's go to B part. Describe the behavior. Again, the word describe or the command word is describe. So please make sure that you understand the command words and you understand how many marks are to be given to you. The things will be easier for you then. Describe the behavior of chromosome during the main stages of meiosis 1. So if you see, you just have to recall main stages of meiosis 1 in animal cells. Okay. In my view, it is a very straightforward question in which you just have to recall the main stages of meiosis 1 and just give at least 1 point for each. If you see, the marks are 4. So 4 means very straightforwardly 1 mark for each terminology. So number 1 is prophase 1. So prophase 1, as you know, there are many things happening in prophase 1 of meiosis 1. Please make sure that you are understanding we are talking about meiosis 1. We are not talking about mitosis. It's meiosis 1, and even we are not talking about meiosis 2. So only the main stages of meiosis 1 we have to deal with. So basically you can write anything, but make sure that whatever you are writing, it should be one of the things happening in prophase 1 of meiosis 1. So mainly the homologous chromosomes pair up, you know very well, they make the synapses. The crossing over happens between the non-sister chromatids of homologous pairs of chromosomes. Chiasmata formation takes place or chiasma formation takes place. You can write anything, any of these this information, but make sure it should be just related to prophase 1, because one line will be enough, or one information will be enough, or in order to save yourself you can write at least 2 but make sure they should not be contradictory to each other. It should be only supportive. The next terminology is metaphase 1. So metaphase, whenever the metaphase terminology comes, you know that the chromosomes, they align themselves on the equator. But here you have to talk about metaphase 1. When you are talking about metaphase 1, you have to say bivalent chromosomes or homologous chromosomes. They line up on the equator of spindles. And definitely, if we are talking about mitosis, we will not be talking about bivalents, we will not be talking about homologous chromosomes, and we will not be even talking about independent assortment of homologous chromosomes. So this is specifically and specially relevant to metaphase 1. The next terminology is anaphase 1. In anaphase 1, the main and typical feature of anaphase 1 is the chromosomes, they start going apart from one another and they go to the opposite poles towards the centrioles. So this is anaphase 1. So your homologous chromosomes, the pairs, they will separate, but the centromeres will not separate. Make sure you make it very clear. So chromosomes, they move towards the opposite poles, but definitely the centromeres, they do not divide yet. The next and the last terminology is telophase 1. In telophase 1, the nuclear envelope reforms, chromosome number is already halved, and chromosomes start to decondense. So any of the information or any of the point you can write over here, you'll be getting 4 marks, 1 mark for each terminology. Okay, so state the term means you just have to write the terminology and that's it. And if you see, I have underlined the terminology cytokinesis. It means this is the only terminology the examiner will be accepting, because this is the correct, I should say the exact terminology, not the correct one, but I should say the exact terminology which the examiner is looking for. So cytokinesis is the term which is used to describe the division of the cytoplasm. So this is for 1 mark, and that's the end of question number 1 and 2 of today's past paper. That was 970042 May June 2023. Thank you so much.